Hi, my name is Alex Collias and I'm a PhD student in the Applied Biology program. I'm going to talk about my research today on the impact of infection with the Helminth parasite Schistocephalus solidus on adaptive immunity in the Threespine stickleback fish. Worm parasites, known as helminths, modulate their host's immunity. And so essentially what this means is that while the host's immune system is working really hard to fight off the parasite is also fighting back in order to overcome those host immune defenses and ultimately remain within the host. So I'm specifically asking how helminth parasites affect their host's immune systems during infection, and I will be using the Threespine stickleback and its helminth parasite Schistocephalus solidus in order to answer this question. So a crucial part of the stickleback's fight against helminths is what's called their B and T cell response, and I specifically want to know if the amount of B and T cells in the stickleback are being suppressed during infection. So I'm investigating this by taking tissue from infected, exposed but not infected, and control uninfected fish. I'm obtaining sections of these tissues and then using a technique that's known as RNA in-situ hybridization to visualize the B and T cells in the tissue. So in this image here, you can see that this is a spleen section and that each small pink dot is a T cell and then each blue spot is a B cell. And then from these images, I can quantify the number of B and T cell spots in a certain area and then use that to determine if infection is actually causing these numbers to change. So this plot is showing the density of B cells in the unexposed control fish, the exposed, uninfected, and the infected fish. So what you see in the y-axis is B cell spot density. And so in the infected fish, you can see that the B cell density is significantly lower than in the exposed but not infected fish. And this suggests that helminths are suppressing the B cell response in these infected fish. This plot is showing the density of T cells in the same three groups. And so while none of these associations are significant, it does appear that there may be fewer T cells in the exposed, uninfected, and infected groups compared to the unexposed control groups, but more sampling needs to be done on these. So in conclusion, B cells do decrease during helminth infection in the stickleback, but T cells do not appear to be affected by infection. And our next steps in this analysis will be to analyze additional samples, perform outlier analysis on some of those outlier points, and also to look at different worm populations. So these studies were done using one specific worm population, and we'll branch out and look at others in the future.